Hello, thank you for coming to the webinar. I'll be talking about a rather specific topic, which is nucleic acid structural quality, not just structures, but quality of nucleic acid structures. And I'll try to persuade you that this is actually important issue. So I work in Czech Republic, in Prague, or almost in Prague, at the Institute of Biotechnology of the Czech Academy of Sciences. So I'll just start with the presentation. So why do we need to talk about quality of structural data? I believe that the public archive, I'll talk about basically what's in the public archive. That's what people access. And in the public archive, they should access reliable data. They should be validated. And in case of structural biology and also nucleic acids, this is PDB, what is public archive, and it's in very good shape, but still it's not perfect, obviously. Why do I concentrate on nucleic acids, and they should be on certain specific concern, and I'll try to tell you why I believe so, not only myself, but I'll tell you some reasons. The number of protein structures is quite large, but number of nucleic acid structures, and especially RNA structures, is still comparatively very, very low, despite huge progress. Okay, this is absolutely no criticism of people who work who solve DNA and RNA structures, but the number of these structures is still comparably very, very low. This is the bars look nicely similar, but notice the logarithmic scale here. And the situation is actually more funny if you concentrate on number of residues, which means amino acids and nucleotides that are available in high quality, high resolution structures. Better, here I talk about better than 2 angstrom X-ray structures. And again, bars are nice, right? But then again, this is logarithmic scale. And if you go to linear scale, you see just nothing at RNA and DNA. It's really about 50,000 RNA and DNA nucleotides, and like 70 or 60 million amino acids. Okay, so we have to be very careful how we treat these molecules. These structures are very precious. Okay, now I'm talking about quality. So what are the sources of bad quality? Okay, what are the source of problems with nucleic acid structures? We deal with valence geometry still. The standards are not always well kept. Base pairing, this is crucial biologically, absolutely essential property, and then backbone geometries or overall geometries. Conformations. Here, geometries wouldn't be. It's not good word. Conformations would be better word. So we report all these properties at our website. So you can say this is a little advertisement, okay? natco.mos.org. You see the address, you can check it. We completely redesigned the website. I'll very briefly go over what's available. And what you see here is the static picture of the homepage, where there is some simple questions, what to upload. You can upload either structures from the archive, your own structure, or structures that were run through PDB redo by Robbie Yosten. And then you push analyze, and you get basic screen with basic information on the left and some Molstar generated picture of the backbone of the nucleic acid in question. What is, I think, in this picture interesting and important is that you get very concise information about the conformation of the molecule, because we have some simple color codes on top. So A-like conformers are in light yellow, B-like in light blue. Then this special B form, which is called B2 form, is in dark blue. Then some mixed, what we call mixed B, strange B-like. So obviously all these B forms are more in DNA, not so much in RNA. Then Z forms, so left-handed forms, and then IC is intercalated. Oops, I'm sorry. Intercalated. There is just one here in the back, a little bit hidden. And then these red guys are so-called open forms. So the bases are not stacked and wide open, okay? So this is basic information about the molecule you get very quickly, and you can catch a hotspot, so to speak, of the structure. So valence geometry, there are some summaries. I don't have time to go into details. You can go into great detail of every nucleotide in the structure, how we think it conforms with the valence geometry standards, distances and angles. We provide pictures, we provide tables. You can always, for all features of the NATCO, you can always download results. Yeah. Okay, next thing I mentioned are base pairing, and this is really essential information from the biological point of view, because we all know about basically coding pairs, CG, AT, but then, especially in RNA world, the non-canonical pairs are very important from evolutionary point of view, and if they are correctly assigned and can be analyzed from the evolutionary point of view, you can use these critical non-canonical base pairs as, say, GA or GU to see evolutionary preservation. Yeah, but unfortunately we discovered that the archive is not very reliable, so we started to develop our new independent base pair assignment. It's based on Leontis-Westhoff classification of these three edges: Watson-Crick edge, CH, or in case of purines, this is Hookstein edge, and then sugar edge that can involve O2 prime in some hydrogen bonding. We have a website that is in development, under development, basepairs.mos.org, that gives you a very quick overview of the assignments based on this Watson-Crick schema. These are these 12 on top of the page. There are these 12 classes, and then you can go to, for each class, obviously there are sequences, right? You have pairs between A and C, and etc. You know, CU, etc., etc. The numbers in parentheses means how many pairs we observe. So in some cases there are none. Yeah, for this particular type, trans WS, which is trans Watson-Crick sugar edge, there is nothing in AU observed. Okay, and then by clicking at the icons, you get the list of all these guys. Say for AA, you get these 39 cases, and you can look at all these things in detail. The third issue is, or the third point of view is nucleic acid backbone geometry. Again, geometry, I have to change it to conformation. This is feature that is often ignored with remarks like, you know, the backbone will always somehow make it or adapt it to the conformation. But we know that the backbone also has only a limited number of possible conformers. Yeah, it's not everything goes, okay? The number of conformers is relatively large, but if you consider the size of the building block we analyze, which is dinucleotide, 96 dinucleotide, these so-called dinucleotide classes, these NTC classes, it's not large number. And more in this line, many of them are very rare. So there are just basically 20, maybe 30 classes that are frequently observed in the structures. So when you go to natco.mos.org, you can get a lot of information, detailed information about the NTC classes. There are several levels of the classification. One is this NTC proper, and then something we called Kana alphabet. It's a structural alphabet that we only roughly classify dinucleotides basically into these colors or into classes labeled by these colors here. So A-like, B-like, Z-like, intercalated. Intercalated means distant bases but parallel, that can be intercalated by something typically aromatic or arginine side chain. Then in these color columns, what you see is how well, the first column shows how well the dinucleotide that is labeled here, named here, how well it conforms to our classification. And if there is 0, it does not conform, it's not classified, but we still see dinucleotide that is closest, or NTC class that is closest. And in this case, I don't know if you, I hope you can read it, it's called AA05. It doesn't matter what it means, but AA is always A-like, and 05 is 5th in the list. If you go here, if you go to browse, you can really look in detail what AA05 or whatever AA00, which is canonical A form, what it means in terms of torsional angles, how many there are, and in what structure, etc., etc. This is the slide that shows validation tool, new validation tool that we developed, and that is very powerful. And again, very quickly shows you where are you in with your structure, or with archival structure for that matter. On the horizontal axis, you have so-called RSCC, which is real space correlation coefficient. So in other words, how well the dinucleotide, because here we are talking about these dinucleotide building blocks, fits into electron density. So you want to be close to 1, yeah, because you know, correlation coefficient of 1 is perfect fit. And then the y-axis, the vertical axis, is RMSD, root mean square deviation from the closest NTC, dinucleotide building block. Yeah. Here you want to see your structure as low as possible. Yeah. So for this, this is for some test structure, and these assigned dinucleotides fit very, very well. Yeah, these are in red marks or crosses or whatever. Yeah, you see that nothing is here, nothing is, you know, you certainly don't want to be here in this area because you have low fit into electron density and you have poor RMSD. Now this is I don't know if I have like 3 minutes. I would go to the website, just show you that it really exists. Here are examples on top. I usually use this one. This is some ribosomal fragment. So it also was used for the presentation. So you see this is RNA, as I said, some ribosomal fragment. You see that even in this structure, there are some bee-like fragments. This in this light blue. In gray, it's what we don't know how to assign it. It's unassigned. Yeah. And again, you see these hotspots in red. Yeah. Here green. So this is a short segment that is left-handed. Yeah. And this is actually quite frequent that in loops there are these left-handed things. There are some, you know, basic information about structure, not so important. Validation. You see always on the left side, you see tabs that brings you to different level of information. Overall quality is what it says, you know, overall quality in some kind of slider view, similar to what's used by the PDB. Backbone quality is the table I showed already with these MTCs and the RMSD from the this is what I forgot to say. This is RMSD again from the closest NTC, but in Cartesian space. This is in torsional space. This is in Cartesian space. This is in angstroms. You can get a lot of information, step torsions. I have to select something, then you get data. A similarity plot is how close your analyzed step, which is this one, is to all other NTCs. You see here it's AA08. I will click here and it's, sorry. I probably don't use it as I should. Sorry. Because this is not AA. Well, I tried this one. No, it works. It works. Okay. I clicked on the red one and it shows this OP is open 13. Yeah, that's the selected one. And you see that it's actually quite unique that all other guys are pretty far. Yeah. This is our RSCC RMSD plot I was talking about, distances and angles. Okay, refinement I'll skip. This is a special issue and I think Markéta will talk about it. And you can download a lot of stuff and you can browse, as I said. Again, on the left, you have different types of data you can browse. Yeah. Base pairing, we would go to the base pairing website specific. I talked about. Yeah. Table of conformers is this detailed table of all these 96 guys. And I think I should not really talk more because my 20 minutes are over. And therefore, I'll thank for your time and maybe if we have questions, I can try to answer them.
Okay, so thank you very much for the introduction. So yes, Bohdan, as Bohdan said that he was talking about the validation evaluation is not about the real structure, so this is gonna be about the real structure, real structures. And it's my great pleasure to be here, and also an excellent opportunity to present my PhD topic for the very last time, because now I'm changing my scientific focus entirely. And so my PhD thesis was about studies of bacterial transcriptional repressors from the SorC protein family, with a special focus on their 3D structures. And I must maybe apologize a little bit from the very beginning, because Bohdan made a kind of advertisement that I will speak about the refinement of the DNA, which I will not, but we definitely can discuss it later during the discussion phase. So I'll start broadly about the project. Bacteria, as all other organisms, needs to regulate the gene expression in order to achieve the best energy balance and to keep their flexibility to environmental changes at the same time. And transcription is the most frequently used platform in bacteria for this kind of regulation, which involves an intricate combination of mechanisms and pathways. And transcription regulators are the most prominent players in it. These regulators are clustered into protein families based on their sequence and structure homologies, and the SorC family is actually one of them. It was named after the first characterized member, a regulator of Cerberus catabolism in Klebsiella pneumoniae. SorC proteins are spread across the bacterial kingdom, and their regulatory functions are known in carbon metabolism and quorum sensing, and both of these domains make them interesting targets for biotechnology and therapeutical applications, as well as the basic research, which was my case. Their protomers consist of two domains, the N-terminal DNA binding domain and the C-terminal effector binding domain, which are connected through a short flexible linker. It was known even before that through both domains, the proteins form tetramers in their apoforms. So far, only one SorC transcription activator has been identified. Otherwise, they are known as repressors that work under so-called switch off and on mechanism, which means that in the case that the products of the regulated genes are not needed, the protein sits on a specific region on the DNA of the DNA sequence, which is called DNA operator, and sterically hinder the RNA polymerase. But once there is a change in the environment, for instance, when the cell is pushed to utilize an alternative carbon source, this change is reflected by the presence of a small effector molecule. This effector binds to the effector binding domain, triggers the conformational change of the protein, and this change leads to decrease of the affinity to the DNA, so the RNA polymerase has a free space to proceed the transcription. The SorC family is identified by the sequence of the highly conserved effector binding domain, which is folded into typical double Rossmann fold, and is further divided into two subfamilies, DOR and CGGR, based on the DNA binding domains. And these domains fall into different type of helix-turn helix motif. As it is typical for basic research, the principal reason why we decided to study these proteins was the knowledge gap. And this table shows all published SorC structures until 2021, which was the year of our first contribution, where we could find only 2 full-length models, both belonging to the DOR subfamily. The rest of the table are dissected or were dissected effector binding domains from which 3 structures were determined in complexes with their effectors. So such a quite limited list of almost 9,000 proteins which belongs to the family. Moreover, there was no structure of any protein DNA complex that might elucidate the principle of the repressor's function and elucidate if there are differences between the subfamilies. And so this was my task, and for which we selected 2 representatives, both from the model bacterium Bacillus subtilis, which these proteins that we selected also gave the name of their subfamilies. These proteins were selected first because they were in long-term interest of our laboratory, and more importantly, they were among the best functionally and structurally characterized. CGGR stands for Central Glycolytic Genes Regulator, and DOR stands for Deoxyribonucleosides Regulator. CGGR regulates genes for enzyme of triose phosphate interconversion in glycolysis, and DOR regulates genes for enzymes of deoxyribonucleoside catabolism as an alternative carbon source. As I said, it's both proteins were among the best functional and structurally characterized. It meant that we knew the pathway they regulate, the operator sequence, and in both cases, the structure of their effector binding domains were determined before, either also in complex with their effector molecules as well as in their free forms. And speaking of the effector, for CGGR, fructose 1,6-bisphosphate was identified as the right one, and for DOR, it was deoxyribose 5-phosphate. And so the remaining question mark was the DNA recognition. It was domain 1, and the additional ones were other ambiguities that we wanted to address, and I will touch during my talk. The key method we used to study them was X-ray crystallography, which was combined with cryo-EM or cryogenic electron microscopy in the case of CGGR. But let's have a look at the full X-ray guide first, and jump directly in the crystallization of its complex with the DNA operator, which consists in this case of 39 base pairs, where we recognize a palindrome in the left part and direct repeat, which is quite asymmetrically located within the right part of the sequence. When crystallizing a protein DNA complex, we must pay great attention to the design of the DNA duplex. And usually we must try many of DNA variants. Is because we deal with the problem of the DNA flexibility, and in this case also with the flexibility of the domains. And so the very quite commonly the solution is found in using a minimal functional region of the DNA just to increase the probability that the molecule will pack in the crystal regularly. And so, although we knew that the whole operator sequence is necessary for the protein tetramer binding, what was suggested before, I tried to crystallize the protein also with the truncated half operators, because at that time we didn't know if the complex is formed through a compact tetramer or by 2 dimers where each dimer binds a half operator. So I tested several DNA variants, and indeed the final winner was the left half operator of 18 base pairs. It was the left part, the palindromic part, that I crystallized in optimized condition by vapor diffusion technique. And the ground crystals, after quite a long waiting time, eventually diffracted to an acceptable resolution of 3.7 angstroms. So the structure could be determined, and here is a built model of the asymmetric unit consisting of two non-interacting dimers, each bound to one palindrome. And this asymmetric unit might indicate that the biological unit of two separated dimers, of two separated dimers might be the right one, but the true biological unit we looked for was found by inspection of the crystal contacts, where we found tetramer formed through interaction of the effector binding domains analogous to the known SorC apoform structures. So this is all beautiful, but this was not the end of the story, as this resolution couldn't answer the question about the protein DNA contacts. And by the way, it would be also hard to evaluate it. So we went for an approach of what I call jigsaw puzzles, where we took the separated DNA binding domain and crystallize it with the half operators. And finally, the complex with the left palindromic part consisting of 15 base pairs diffracted best to the high resolution of 2.3 angstroms, and we could integrate it into the full model. Moreover, this structure allowed us to predict the contacts along the wall operator sequence. Even along over the right part. The relevancy of the tetrameric unit was confirmed by the size exclusion chromatography, which also showed that the elution profile of the protein DNA complex does not change upon incubation with the effector, which indicates that the protein remains attached to the DNA even upon binding of the effector molecule, the deoxyribose 5-phosphate. So this was great. However, what was not confirmed was that the sole left half operator is not sufficient, or it was not confirmed that it is sufficient for the complex formation. One of the methods I used to tackle this was microscope thermophoresis, that showed that the direct repeat of the operator is needed for the protein binding in solution. So we had to ask how it is possible that we crystallize the complex with it. The answer was offered by the combination of mass photometry and size exclusion chromatography that showed that the protein exists almost exclusively as a tetramer in a wide range of concentrations. So it is highly plausible that in a crystallization experiment where both components are in a higher concentration, the prepared tetramer cut its binding pattern easily, and the complex was additionally stabilized by the crystal environment, by the crystal contacts. Anyway, Zhang and his co-workers, who published the characterization of DOR in 2000, they were right about the DNA sequence binding. On the other hand, methods we employed contradicted other assumptions they made. First, we found that DOR probably binds the DNA with 3 or the lower affinity, and the binding is not demonstrably cooperative as they claim. So by this, I'm leaving DOR, and let's continue with the second guy, CGGR. Where we combined X-ray with cryo-EM. CGGR has been extensively studied by a variety of biophysical methods before. We knew that the protein binds DNA as a tetramer in a highly cooperative manner, having almost 100 times higher affinity to the right half operator. We also knew that upon its upon binding of its known effector, fructose bisphosphate, the tetramer disintegrates to two dimers, which, however, remain attached to the DNA. And finally, the rough model of its complexes from small angular X-ray pool was also published. And surprisingly, this model showed the protein DNA complex as such an asymmetric particle, which made us ask if it's for real, if it's really can be that the effector binding domains don't interact with each other like within the wall complex. And to tackle this problem, we employed cryo-EM. Here, just to balance the crystallization part, in the case of DOR, I just want to show the importance of doing sample quality check before you go for cryo-EM itself. The check is done by negative staining of the sample and analysis by transmission electron microscopy. And it can clearly show you that your purification protocol is way far insufficient, as it was apparently in our case. So with this optimized protocol, we got a high quality protein solution and could continue with cryo-EM. And in the course of this experiment, we selected over 220,000 particles to reconstruct the 3D cryo-EM map of a global resolution of about 4.3 angstrom, which clearly shows a C2 symmetrical complex where all the effector binding domains are in contact. So we make the previous sex model much more accurate, which is just this complex is in contrast to the sex model. Nevertheless, we encountered the same problem as in the case of DOR for the low resolution we got from the method. So to build the effector binding domain portion, we could use the previously published crystal structures, but the DNA binding part was invisible for us. And so the jigsaw puzzle game took place once again. We crystallized the separated DNA binding domains with half operators. These crystals gave us a resolution of 2.1 angstrom, and analogous to the previous case, it allowed us to complement the cryo-EM model. Another question which remained unanswered for a very long time was the promiscuity in the effector binding. The previous study presented several crystal structures of liganded effector binding domains in whose ligand binding sites were found several glycolytic intermediates. So besides fructose bisphosphate, there was also dihydroxyacetone phosphate, DHAP, but also glucose 6-phosphate and fructose 6-phosphate. In the same study, they also did isothermal titration calorimetry, which showed that DHAP had as high affinity as FBP, fructose bisphosphate. Nonetheless, its real effect on transcription was not studied further. So we asked this question, and first, the in vitro transcription assay was applied to test the effect of the suspected ligands, and it really showed that DHAP induces the transcription even more efficiently than FBP. Further, we wondered if we can capture the disintegration of the complex upon the addition of the effectors. And if we do, then if we will observe the same for DHAP and FBP. For this, we used cryo-EM again, and the 2D classes clearly show that the effect of FBP and DHAP make the complex disintegrated either completely or dissociated either completed or only a dimer remains attached to the DNA. In contrast to the protein DNA complex, or in the presence of the ligand that didn't induce the transcription, where the particles remains intact. So the conclusion for CGGR is that it indeed forms symmetrical tetramers. We could reconstruct its interaction with the wall operator thanks to the high-resolution crystal structure, and we confirmed the second effector dihydroxyacetone phosphate. And let's conclude this talk by putting the SorC siblings side by side and compare them a little bit. We showed that both representatives form tetramers of a very similar shape. However, their architecture, which is dictated by positions and compositions of their DNA binding domains, is different. In DOR, we observe domain swooping where the monomers cross each other, but this is not the case of CGGR. I don't have time to go into details, or didn't have time to go into details about the protein DNA context, but what I would like to highlight here at least is that both proteins contact their recognition bases by highly conserved arginine residues, which indicate that this might be the case, or this will be the case for the whole SorC family. And finally, so let me thank all the contributors to the project, without whom I would not defend. Thank you for being here with me, and now I will do my best to answer your questions if there are any. Thank you.